Introduction. Blastocyst embryo biopsy is a laboratory procedure that provides pre-implantation genetic testing on embryos by collecting cell samples when they are at the blastocyst stage. In other words, at day 5 to 7 of development. Fertilization occurs when an egg and sperm fuse together to form a zygote. The zygote is a single cell that continues to divide to form a spherical layer of cells called a blastula, which comprises roughly 100 cells. The blastula contains a fluid-filled cavity called the blastocele. Each cell of the blastula is called a blastomere. As the blastomeres continue to divide, they form a blastocyst, which contains 100 to 200 cells. The blastocyst contains an inner mass of cells and an outer layer of cells called the trophectoderm. The inner mass develops into the fetus, while the trophectoderm develops into the placenta and the other supporting structures during pregnancy. A blastocyst biopsy gives better accuracy and more reliable results in genetic testing as we are sampling more than just a single cell. Indications. Blastocyst embryo biopsy is indicated for genetic disease prevention, to increase the chance of a successful pregnancy, sex selection, and other reasons. Embryo biopsy procedure. A sample of embryo on day 5, 6, or 7 of embryonic development is taken. The sample is stored in artificial media that mimics the human uterine layer. It is allowed to incubate to facilitate expansion of the blastocyst. After the cells have matured, a minute hole is created in the blastocyst sample using a non-contact laser. A few blastomeres are collected by applying gentle suction with a biopsy pipette and transferred to individual test tubes or petri dishes which have a medium to keep them nourished and viable. The medium contains PCR-grade water, mineral oil, and nutrients. PCR-grade water is specially purified, double-distilled, deionized, and autoclaved water. The cultured blastomeres are subjected to a genetic test known as polymerase chain reaction, PCR, to determine if they have the proper chromosomal composition. PCR testing is performed to replicate the DNA in the blastomere. The DNA samples are tested for protein markers that help identify any abnormality. The embryologist or fertility specialist will vitrify or flash freeze the tested embryos and store them safely until it is time to transfer the embryo into your uterus. Following the biopsy. Your doctor will schedule your next appointment soon after the results of your genetic tests are available. Risks and complications. The biopsy procedure is safe, but does require careful planning and collection of the blastocyst from the embryo. Possible risks and complications with a blastocyst embryo biopsy may include improper selection technique leading to damage to the embryo, inaccurate results. Summary. Blastocyst embryo biopsy is a diagnostic procedure to detect genetic abnormality during the blastocyst embryonic stage. Post-fertilization, the cell samples are collected when the embryo is at day 5, 6, or 7 of development. After testing, the healthy embryo is transferred back to the uterus for normal development. Blastocyst embryo biopsy is considered a safe procedure and does not adversely affect the growth of the embryo.